Hi, everyone. Thanks for joining me here today. It's a real pleasure to be able to present this paper to you, and I hope you find it interesting. My job today is really to talk about the ethical aspects of CRISPR, but I'm not going to focus on the kind of more traditional or standard ethical questions around safety and risk and so on. I think those are interesting and important questions to answer, but there is already a large discussion going on around those things, and some of those are kind of technical questions that I can't answer as an ethicist. So what I want to do is actually look at CRISPR within a broader context, and I'm particularly going to be talking about using CRISPR for germline modification. So I'm going to be talking about it in the context of human reproduction. And I want to think about what CRISPR will do in terms of how we think about ourselves within that broader context. So what I'm going to do in my paper, hopefully, is not answer the question, is the technology permissible or not? That seems to me, as I said, an important question, but not one that I'm going to answer in this paper. Instead, I want to take a slightly broader focus and ask what social and ethical effects might this technology and its use have? So we might ask questions like, what future does it make possible? And what future does it close down? What are the opportunity costs of actually using CRISPR to do germline editing of humans? What might it make us? How might we think about ourselves if we do use germline editing? Or what ethical relationships, what kinds of subjectivities or intersubjectivities does it foster or erode? So broader questions than just about risk. So just to give you a sense of the structure of the paper, I'm going to talk very, very briefly about what CRISPR-Cas9 is, and then talk at more length about how we might put CRISPR in its context of innovation in human reproduction. And try to get at why that's important, I want to look at three or four groups of people or objects that actually matter for thinking about what CRISPR does. So I'm going to specifically talk about scientists, women, children, and the technology itself. And then I will return to my broader questions about ethical futures to conclude. So on to the first section, what is CRISPR-Cas9? I know for this audience, I don't really need to give much attention here. So I will just very briefly say that CRISPR-Cas9 is often described as a kind of pair of molecular scissors that allows for a double-stranded break that you can then delete or insert a gene to actually change the genome of a human being. Of course, it can be used both for somatic and germline gene editing, somatic being related to the body cells of any particular individual, the germline being the kind of editing that would happen in the context of human reproduction, where any changes made are then inheritable by the future generation. So just to give you a very short history of CRISPR-Cas9. So CRISPR itself was discovered in 1987 by scientists at Osaka University. And in the ensuing 20 years, there were a lot of important work done and a lot of important discoveries made. But the important breakthrough from the point of view of germline editing was the discovery or the refinement in 2012 of the CRISPR-Cas9 system by Jennifer Doudna and Emmanuel Charpentier. That refinement gave rise to, as you know, a lot of issues around patents, an ongoing debate and legal battle around patent issues. It also gave rise to various calls for international moratoria on germline genome editing, including from Doudna. So at the moment, there's still no international consensus on the safety, efficacy, or acceptability of using CRISPR-Cas9 to germline genome edit human beings. Even so, you will know that in 2018, there was an enormous controversy when a Chinese scientist announced that he had, in fact, used genome editing in the birth of two two baby girls in China. And more recently, in 2019, a Russian scientist also announced plans for more CRISPR babies, saying, "I think I'm crazy enough to do it," which is hardly reassuring. That's the kind of situation we're now in. No international consensus, and yet early adopters of the technology already taking it up and using it in human reproduction. So for me, I think it's really important to think about what effects that early adoption of the technology will have in how we think about the ethics of germline genome editing and whether we go forward with it or not. So you will know the Chinese controversy arose around a Chinese biophysicist, He Jiankui, when he announced in 2018 via YouTube and at a conference in Hong Kong that two girls had been born following the use of CRISPR. He used CRISPR to target the CCR5 gene, which is known as an HIV infection pathway. He did this because the biological father of the girls is HIV positive. However, standard practices for preventing paternal transmission of HIV in conception were used in the IVF that was necessary for the birth of those girls. And after those standard procedures, there's really no risk of paternal transmission in conception. The case was controversial for a number of reasons. I mean, one, because the parents were provided with free IVF if they agreed to CRISPR, raising some serious questions around consent and inducement and so on. It's also been controversial because it's emerged that several scientists in the US and UK knew that Jiankui was planning to do this and didn't do anything to prevent it happening. It also seems that the standard processes for ethical clearance were not followed, and there's quite a bit of obscurity around exactly what happened in that case. So the ensuing debate around the controversy has focused on concerns about safety, the therapeutic necessity of the intervention, given that there are already well-known and efficacious ways of preventing HIV transmission, and the transmission risk from the father was minimal. And there's also significant concerns around the consent procedure and questions of inducement. Now, I think those questions are all important and necessary to consider. But what they do is narrow our focus onto that particular case. And when they do that, we lose the broader focus and in terms of thinking about what the uptake of CRISPR actually did in that case. So what we can say is that He Jiankui took away the possibility of a world without genome-edited children. That is simply no longer possible now that we have two gene-edited children in existence. And the fact that we have those two children means that it's much more likely that we will also have more. So in a sense, He Jiankui forced us into a particular future and closed down the opportunity of another without any kind of global consensus around whether the futures were desirable or not. So I think it's in that frame that we need to think about what the early uptake of CRISPR-Cas9 might do. So in order to actually get at this question, I want to take a step back and put CRISPR in context a bit more. Because while on the one hand, what He Jiankui did was quite radical, on another hand, he's actually following a fairly well-worn pathway, which is the pathway of early uptake and innovation in human reproduction. So with frontier scientists kind of using a technology before there is any consensus about whether that technology is safe or not. So that's the context I want to look at the use of CRISPR-Cas9 in. So just to kind of give you a sense of that history and the context that I'm talking about. What I have in mind is the history of scientists and their breakthrough babies. So frontier scientists kind of stepping ahead of international debate to actually use technologies to produce children. So what I have in mind, we have here images of Robert Edwards and the first test tube baby with the advent of IVF in 1978. John Zhang and the first three-parent baby using mitochondrial replacement techniques. That baby was born in Mexico in 2016. And then He Jiankyu and the first CRISPR babies born in 2018. Now, these pictures are just taken off the web. They're fairly random, but there's one striking feature of these, and that is that the mother is nowhere to be seen. Now, of course, there are good privacy reasons for that, but I think it's also a kind of interesting semiotics to note that we have scientists and their breakthrough babies, and the mothers who actually are necessary. We still require pregnancy for babies, for instance, obscured within the thinking of what's actually happening in the advent of reproductive innovation. So I guess the question, one of the frameworks that we can use to answer the question of how we get from A to B is a framework of responsible innovation, or better yet, responsible translation, because it's not really the innovation itself that's problematic, but the way in which innovations are taken up within the clinical context of human reproduction. So it's that translation aspect that's particularly interesting. So how do we think about responsible translation? Well, when I've been digging around in the literature on responsible translation and innovation, I came across one paper that I found particularly interesting. And it's interesting because it really stands out of the concerns around safety and so on. And this paper argues that responsibility in translation or innovation is necessarily responsibility for the future it helps to create. It claims that responsibility for innovation is a condition of mutual vulnerability because of the kind of creatures we are. So the claim there is that by virtue of being human, we are vulnerable. That's the nature of what it is to be human. And you can think about this in terms of the fact that we're all vulnerable to illness, to accident, to misfortune. We're vulnerable to each other, and so on. So vulnerability is a key feature of what we are as humans. So this framework is suggesting that the key terms for thinking about responsible translation are actually things like futurity and vulnerability and intersubjectivity, the kinds of relationships that we have with each other in the context of translation of innovative technologies. So to get at why it might be useful to actually think about those terminologies within responsible translation, I want to actually look at the various actors or actants that we might identify in the process of translation and think about what they do and how they might be affected by the translation of innovative technologies. And the first of these is scientists or clinicians. Now it's, oh, there we go. So the question is, why do they do what they do and what happens to them when they are these kind of front-runner scientists in particular? I think the first thing to note is that scientists, frontier scientists in particular, are world makers who operate in a multidimensional context. And by world makers, I mean that they actually contribute to shaping the life world or the shared life world that we operate in as human beings. They shape our social context in important ways. Now, there are, of course, a number of different pressures operating on scientists that motivate them to act in particular ways. There are banded scientific and institutional pressures, the necessity for impact, the importance of getting grant funding, and so on. So all of those factors actually contribute to shaping the motivations of scientists themselves. There's also the broader context of the precarity of research science employment. I'm not sure how it is elsewhere, but in my context, the employment as a research scientist in a university is often very dependent on research funding, for instance. So there's a certain kind of precarity involved in science employment. increasingly, I think there's also significant commercial drivers operating in science. So this is, if we go back to the history of CRISPR, the patent battles that emerged around the refinement of the CRISPR-Cas9 system are very indicative of the importance of commercialization and the capacity to monetize any invention and so on. So those are really strong motivators, I think, in the context of science today. There are also, of course, just the personal motivations of individual scientists. It may be that some scientists are just driven by the desire to be the first. It may be that they are driven by other motivations, like the desire to please their father. We don't necessarily know what individual motivations are, but they are certainly there and part of the picture of this multidimensional context. I think one thing to note about this context is the way in which the role of the scientist has become merged with the roles of clinicians, but also of entrepreneurs. Scientists themselves are increasingly required to be entrepreneurial. And you can see that kind of strong entrepreneurialism coming out with some of our breakthrough scientists that I mentioned before. So John Zhang, for instance, is very much of this kind of scientist, clinician, entrepreneur type figure. And we also need to think about what risks, what rewards, and what repercussions there might be for scientists acting right at the frontier of translation. So certainly there were some risks for He Jiankui, and some of those have been borne out in terms of the after effects of his use of CRISPR. But presumably there may also be some rewards, or at least he's motivated by the perception that there are significant rewards to be had from being that kind of front-runner scientist. So the point there in relation to scientists and clinicians is that it is a multidimensional context with a lot of different pressures acting on scientists, motivating them to be right at the forefront of translation. Now, what about the women? As I've said before, they're really not part of the picture of the scientists and their breakthrough babies, but they're still essential to scientific innovation and translation in the sphere of human reproduction. So here we need to think about who the women are and what they do. And here I think we can say that women lend themselves to the kind of translational process by giving these early adoption of technologies like CRISPR a certain kind of maternal authority. And sorry, Tracy, my computer is sending me little alarms.
That's okay. I'll make a note of that. What slide? Let's see, you're on slide 16. Okay. No problem.
We'll take that one again.
All right. Sounds good. Thank you.
Yeah. So the second group of people we need to think about are women. So women are really central to the process of scientific innovation and translation today, especially in the sphere of human reproduction. They're still necessary because we haven't yet got past pregnancy. And they're essential because it seems to me that the involvement of women in this process actually lends some legitimation and authority to it. So we can say that there's a kind of maternal authority in the fact that a woman has herself agreed to use this technology to have her children. Now, interestingly, an anthropologist by the name of Rainer Rapp came up with the idea of moral pioneering in reproduction some years ago in the context of thinking about prenatal testing technologies. But I think we can see that this concept is really pertinent here because women who are using these technologies are really right at the forefront of the kind of moral debates of whether these technologies are acceptable or not. So they're very much kind of moral pioneers in terms of the translation of innovative technologies. So it seems clear to me that women are actually part of the legitimation process for innovative technologies. But here I want to be clear that women are neither hoodwinked nor intentionally lending themselves to legitimate these technologies. Instead, I would say, using a quote from the French philosopher Michel Foucault, that people know what they do. Frequently, they even know why they do what they do. But what they don't know is what they do does. And that's a very cute way of saying that people don't know what the consequences of their actions may be, or what the effects of their actions may be. So the last point that I want to make about women is that maternal and parental desire itself acts as a driver of innovation. So we can say this because scientists often point to the importance of reproduction and the strength of that maternal or parental desire in terms of legitimating their use of technology. But we also need to note that that desire itself is not straightforward, because the desire and the technology are themselves co-produced. The desire is not pre-existent to the technology. Nobody's sitting around 10 years ago saying, I'd really like to genome edit my child. It's only when the technology becomes possible that the desire itself becomes pertinent and actually starts to drive that process of translation. So what about the children? Who are they and how do they live? So interestingly, when we talk about the children of innovative technologies, there are two things that we need to keep in mind. On the one hand, what we're talking about is the figure of the future child, the child not yet in existence, but who might come into the existence if we are able to use these technologies. So in that sense, the child is a kind of proleptic target of responsibility. They're a target of responsibility that does not yet exist. But we should also keep in mind that we're talking about actual children that will exist or do exist. So if we think about the history of IVF, for instance, we think about the children who have been born of that technology, and in particular, the first child born of that technology. What is it like then to be a breakthrough baby? What kind of life does one lead as a breakthrough baby? Now, Louise Brown, the first child born of IVF, often kind of downplays the importance of or the effect of being a breakthrough baby on her life. But we might note that not everyone has their 40th birthday at the British Museum and has it broadcast in the international press. So there's clearly some difference to the lives of breakthrough babies. There might also be different moral responsibilities that the children of innovation have by virtue of their experimental birth. Would they, for instance, be obliged to submit to ongoing medical testing, to making their lives public and so on? Because that's how we will know if these technologies are actually safe or not. We could suggest that perhaps they have an obligation to not reproduce, because really that's the only way that we will be sure that the technology is safe. We won't know whether it's safe for the next generation or not until that next generation comes along. So we might also say then that they're in fact obliged to reproduce, because that's the only way that we will know that the technology is safe. So it may be that the children of experimental births actually have certain kinds of responsibilities or obligations by virtue of that birth. Now, the last thing I want to talk about are the technologies themselves. What do the technologies themselves actually do in the process of translation? Well, there are a few things to say, but the first of these is that technologies make possible other technologies that are neither intended nor foreseen. So if we think about in vitro fertilization, for instance, no one imagined when that was being developed that it would give rise to other technologies such as pre-implantation genetic diagnosis. That was just not part of the picture, and yet PGD is only possible because IVF is possible. We can also say that technologies have social effects that may not be foreseen or intended. So using IVF as an example again, no one foresaw that IVF would give rise to a system of international commercial surrogacy, or could give rise. And yet that's what it's done. It's made possible certain kinds of international commercial surrogacy arrangements. So what also comes from that is that technologies make some forms of social existence possible and put others into question. So when we think about IVF again and gamete donation, that process of IVF and the possibility of using donated gametes has really put into question the importance of genetic relatedness in a way that 40 years ago just wasn't questioned at all. Now, I'm not saying that prior to IVF, all families were based on genetic relatedness. That's clearly not true. But the way in which we thought about genetic relatedness was very different from how we think about it now. So given those things, we think the question to ask is, what might genome editing do? What kinds of other technologies might it give rise to? What social effects may it have? And what forms of social existence may it make possible? And what forms of social existence may it render impossible? In other words, what are the opportunity costs of using CRISPR-Cas9? Because everything has opportunity costs. Any avenue of action will have opportunity costs. So what are the opportunity costs of using CRISPR-Cas9 for germline genome editing? So this takes me back to my concluding slide, which is, and returning us to the question of ethical futures. So I want to emphasize that the point that I've made is that the context of translation in human reproduction is very multidimensional. There are a lot of different pressures coming into play. There are a lot of different actors and actants with different effects and implications and power and so on. I haven't given an exhaustive list of what those actors are here. I've just given a small selection of what they might be. We also might think about institutional review boards, for instance. What effect do they have? What do they do within the process of translation? But I want to, again, push home my central point that responsibility associated with innovation is necessarily responsibility for the future it helps to create. In the process of taking up any particular technology or using a particular technology, we create certain futures and render others impossible. And this reminds us of the centrality of intersubjectivity and vulnerability in the process of translation. Translation is about a shared world as much as it is about individual desires and interests. So in relation to genome editing, I think we must think what it is that we are doing. That is, what world are we creating for future generations, not just for us, but for our children and our children's children. Thank you.